Plant Sciences Symposium. My name is Elizabeth Blissett.
And I'm Austin Dobbles.
And we are the co-chairs of this year's Symposium Planning Committee. So we, along with our planning committee, have had the honor to plan and host the 11th annual symposium event being held here at the University of Minnesota. Our two-day event began yesterday with an outstanding workshop where we had the privilege to hear from expert panelists on how quality matters beyond plant sciences. This was followed by Dr. Bernardo's Plant Breeding Games competition, which is a Rex Bernardo original simulation-based tournament where teams competed to successfully develop a barley cultivar that met craft brewing needs and quality standards while maintaining yields.
Yeah, to follow up on that workshop, the winning team produced a barley cultivar that yielded 119 bushels per acre while meeting quality standards. So it was really challenging to meet those quality standards, but it was a lot of fun participating in that part of the workshop. That workshop actually segued into today's event. Today's main event's theme is centered around breeding for end-use quality, with the theme of From Markers to Markets. We have invited 5 scientists from across the country to discuss their background and share their insights on this topic.
So this year in particular, Austin and I have been incredibly grateful to have had the opportunity to lead our team of 14 dedicated and extremely hardworking graduate students to plan this event. The student-led nature of the Pioneer Symposia series is something that makes it truly unique. Planning for this event started over a year ago, and since that time, each student on our committee has taken on their own roles and their individual responsibilities and worked together to make this event happen. So we would like to take a second to sincerely thank every member of our planning committee.
We are also very fortunate to have received funding from the Minnesota Soybean Research and Promotion Council to help fund our student travel grants. We were able to bring in 10 traveling students from across the United States from neighboring universities to attend both our workshop, our dinner last night, and our symposium today. We're excited to have the opportunity to engage with them and learn about their research. You can find out more about their research during our breakout sessions later today, as well as the poster session, which is down the hall in the Cherrywood room. I'd also like to recognize the funding we've received from the College of Food and Agriculture and Natural Resources Sciences, the Scholarly Events Grant, which helped cover the cost of this facilities reservation.
We would finally like to thank DuPont Pioneer for their 11 consecutive years of funding, and especially Tabaré Abadi for his continued guidance and support for each planning committee throughout the years, but especially to our own. This event would not have happened without their support, and especially Tabaré's generous support and expertise in guiding us along the way.
We'd also like to point out that this symposium is one of a series of many flagship by DuPont Pioneer in collaboration with many universities internationally. And we'll welcome Tabaré Abadi to the stage, and he's going to tell you more about the series as a whole. So sit tight, and I know he's got some really cool slides coming up, so please enjoy. Also, if you are tech savvy and on Snapchat, we have a filter, so snap it to your friends, add it to your story. And we also have Twitter and Facebook, so if you're on your phones, I expect high social media engagement. Let's welcome Taba.
Okay. I think I can do it from here. It might even be better that I do it from here because it's actually a little bit easier. I'm Tabaré Abadi, and I work for Corteva Agri-Sciences. Okay, I mean, I wanted to introduce the name because it's a name that you guys are going to be listening in the future, and I'm not going to make a big deal about this at this point, but if you have questions about why the name changed and anything like that, please come and talk with me or with any of the other Corteva Agri-Sciences scientists that are here today. So a little bit of, first of all, thank you everybody for being here. I mean, you know, as Liz and Austin said, you know, this is the 11th consecutive symposium here at the University of Minnesota. And It's been an honor to be part of this story from the very beginning. And but I think that, to be fair, I have to put this story a little bit in a context, right? I mean, because I want for you to understand what has been, I mean, what has been the whole story of the Symposia series. So all this started actually here in this campus, and it started in 2007 with a letter that we received, that actually Jeff Graham received from Rex, that is here sitting in the front row, and I am pleased to see that Rex is here too, because this letter was an invitation to sponsor for 3 years a symposia series organized by grad students, right? At that time, the first 3 years, I think at least the first 2 years it was in the Cargill Center. I mean, it might have been even the first 3 years. And some of you were part of the organizing committee. Vance was part of the organizing committee, and I think that John might have been part of the first or the second, and I'm sure that there are others of you here that have been part of that story too. So it was a very successful story. I mean, the students did a great job and led fantastic events, managed the logistics, and we saw people grow a lot in the process, right, and learn a lot of skills. So now this is what we have, okay? And so the idea was that this was a successful story. Why not take it to other places? And of course, we didn't go from Minnesota to all these other places on a single year, right? I mean, it was all these other 9 years or 8 years in difference, but we have grown a lot, right? And the idea is that, this is a great opportunity for development of skills for graduate students, a great opportunity to develop connections around the world, and a great opportunity to enhance career possibilities for future scientists in the area. So the basic original example or idea, is now being applied everywhere else in the world. I would say, I mean, it's not 100% everywhere in the world, but you might have seen that, you might see here that we have strived to actually take it to as many continents as possible. And the program is still growing, actually. We are still having requests from other parts of the world. This week, we had a request from Erie to join the series. And of course, we said yes. We want to go to Philippines, and it has been a great adventure. Last year, Rex was in Paris, right? And the picture in the front is a picture from the Paris Symposium, and Rex said himself, I mean, how could he have imagined that when he wrote that first letter that we would be, taking, or bringing opportunities for people to connect around the world in this way, and especially for grad students around the world to connect with each other. So what is this series, okay? And so all these events around the world are joined in common objectives, right, that are to contribute to education, to contribute, to do outreach, and to provide career opportunities. The events are fully student-run, and we try to emphasize the component of education in the whole program. We know that it's going to be a great academic event, but we want the students to focus on what they learn on the process. It's more like the road to the symposium. What is the most important thing that we think that we are contributing for them? Another thing is that the students are completely free to select the speakers and the topics, and and again, we assume that in all the conversations on invitations, the topic of talking about relevant science is the main point, and also in most of the cases the events are free. And I say most of the cases because in some cases actually the students have had to put some control on the size of the audience, right? Because in some places it is not so easy to get big places or big venues, and they really need to get some control on the people and assure that there is a commitment for the people that register to participate. So I want to emphasize a few of the major dimensions of the series that I think that are important at this point. One is the global dimension, right? I mean, as you see, we have not only reached several continents around the world, I would say almost, except for Antarctica, we are almost in I don't know if there is summer in Antarctica, we might go, but I don't. But somebody else will. But we have done those connections, and I think the important thing is we have not only have connections done regionally, but we have done connections intercontinental connections too, and we are fostering those things. If we do a zoom in North America, you could actually see how those connections are the history of the connections in North America, and you see that the larger circles are the places where we have events. All the other places are places that have contributed with speakers to the series. So it's not the only way to do connections, but really a lot of connections can be built around this series. Another component is the component of education, and just to highlight what are the things that students take control and on these events, format and purpose, sponsorship and budgeting. I mean, you heard from Liz and Austin, all the sponsors. The students have become very efficient on actually getting additional sponsorships, and we welcome that. I mean, we think that's a great experience. And of course, timing, topic, speakers, communication strategy, development of web pages, video streaming, poster competition, and last but not least, the effort that has been done across North America for student exchange across symposia. And as Liz and Austin emphasized, there are many students from other universities here, and that has become a tradition for most of the events in North America, and we are trying to take this idea to other parts of the world too. Another level is the career exposure, right? I mean, and one level is, how many speakers have come and where are they from, and, if you see in this cloud, these are 400 different speakers representing more than 250 institutions, in 21 countries. So, the series has given the opportunity for students to actually connect with high-level speakers or speakers that are important for them, and then we assume hence that they are high-level too, so that they can get exposed to different opportunities from a disciplinary point of view, but not only from a disciplinary point of view, but also from a career perspective. And we wanted to emphasize here how many speakers have been from academia versus speakers coming from other areas, right? And we have speakers from universities, from international centers and institutes, from government, from nonprofit, and also from other companies outside, that are not pioneer. And one thing that we want to emphasize is that we do welcome that, right? I mean, this is not, a pioneer in the past or Corteva in the future event. I mean, it is your event. We more than welcome you to bring different ideas and to bring to the table, different perspectives and views that we can actually enrich from listening to. And another component is the component of outreach. And actually, we have just begun to to gather this information, so I am only going to share these two examples. The example of the Production Agriculture Symposium here in Minnesota that happened in February, and the example of the ISU, Iowa State University Symposium that happened a few weeks ago in Ames. And the idea is that these events have become a lot broader in audience than just graduate students, right? And there is in all the cases, at least 50% of the participants are coming from outside, the original population of graduate students involved. Whether they are graduate students from the same or from other universities, there are a lot of professionals, people from industry, from government, undergraduates that actually join the events. And so it's a great opportunity to reach out with new ideas and to and to take new ideas to a broader public with this. Just to emphasize one important thing here is in terms of communication, we have two major venues of communication. One is we have a Facebook page that I hope, that Matt Brisman, that is also sitting here in the first row, manages. Matt is a grad student in ISU, and so it's a peer-managed Facebook page that we try to use it as a format of free communication between graduate students. Now we've heard so many things about Facebook, so we will ask Matt to actually give us assurances that he's not selling the information to anybody. But I hope, Matt is from Minnesota, by the way, so I don't think he would. But and then the other page that we have is our own page that is a very, very humble page in which the only thing that we want to share in that page is an open page for everybody to go see, and we have the links to all the individual events that are happening in the near future. We don't have all the historical events, just the ones that are happening in the near future, so that everybody that wants to find out which are the future events, they can either do it through the Facebook page or through our page. And in most of the cases, the individual web pages of the symposia connect with the other symposia also in the series. Last, I wanted to share something with you. We are planning an event in Johnston later this year, in the middle of the year in July. And this is an event that is essentially going to be around genetic evaluation. I would say, the broader topic of quantitative genetics applied to plant sciences. And, we are obviously going to use our venues to communicate about that event. And there are going to be invitations that are going to be sent. And there is going to be a session specifically dedicated for grad students, too. So please be aware of that. And, we would really like a lot of you to consider participating on this event. So before finishing, I want to say one more thing. There are two symposia here, annual symposia now in the University of Minnesota, okay? And one is the Production Agriculture Symposium, and the other one is the Plant Sciences Symposium. And these originally, were addressing or were identified with different groups of students in the department, in the broader area. One thing that I am very pleased to see is that along the years, these two groups have been working together, collaborating, and actually trying to produce a coordination that makes us all very proud. And I think that you guys complement each other very much, and I congratulate you guys for all the achievements that you have had. And I encourage you to keep doing that and to keep working together, because that is the nature of what we want to achieve with this symposia, right? I mean, maximize collaboration, connections, and and working together so that you guys are better prepared for future long careers that you guys have ahead of you. With that, I will thank you everybody for being here and enjoy the event, and hopefully I will be around next year too.
Yeah, we hope to see you next year, Taba, definitely. Thank you again. We'll now bring on Nikki Mihalik to the stage, and she will introduce our first speaker.
Hello. Oh, great. Good morning, everyone. Our first speaker is Dr. Tom Clemente. Dr. Clemente is a professor at the University of Nebraska, where he serves as the director of the Plant Transformation Core Research Facility and holds joint appointments in the Department of Agronomy and Horticulture and the Center for Plant Science Innovation. His research focuses on plant genetic engineering. Dr. Clemente comes to us today to share his research on improving soybean oil composition for value-added output traits. Please join me in welcoming Tom Clemente.
Thank you. Thank you. It's a pleasure to be here, and I appreciate the invitation. It's especially gratifying to be invited by a student-based selection committee. Okay, I think up and down, I got it, yeah. I visited the state of Minnesota many times over the years, and what I really appreciate about this state is the university has a stellar history in the plant sciences, and Bob Dylan from Hibbing. What I don't like about the state is the Vikings. When that highway sign says, road to the Super Bowl to the right, they seem to make a left. So what I put here is an out. Oh, I guess you can't see that well. I should have done a little bit better. Anyway, what I hear is the presentation outline. I'm going to quickly give you an overview of what we refer to at the University of Nebraska as the agricultural biotechnology pipeline. And then I'm going to give you a quick overview of some of the in my facility, we primarily work on 3 crops. That's the soybean, sorghum, and wheat. I'm just going to give you a highlight of the targets that we're going after in those 3 commodities. And then the vast majority of the talk is going to be first discussing designing a soybean oil for baking applications, trying to get an oil that's like a semi-solid at room temperature. And then second, I'm going to give you an overview of where we're at in the status of developing a soybean-based feedstock for aquaculture, trying to get rid of the fish meal and fish oil and farm-raised fish and replacing it with terrestrial-based ingredients. In this case, we're focusing on the soybean. And at the very end, I'm going to give you what I refer to as the Clemente commentary. So, as Nicole alluded, I oversee for the past 22 years a core research facility at the University of Nebraska-Lincoln, referred to as the Plant Transformation Core Research Facility. And what this does is, there's a lot of public sector and private sector transformation facilities, but in the public sector, what really differentiates us is not the breadth of the species that we work with, mainly those commodities, including corn and a couple model plant species, tomato and tobacco, but we have the wherewithal to get stuff out of the greenhouse, and we have dedicated acreage that could be tested solely for regulated materials. And then working with our folks in food and feed science and our industrial ag processing center, we have the capacity for downstream processing of those biologicals if we're working with some output traits. And it's the food and feed science capacity that we work closely with in our aquaculture capacity, because we needed extrusion of the oil to test in the novel feeds. And it's this infrastructure that could take us from the lab bench to the greenhouse to the field to downstream processing in a strictly identity preserved fashion that we refer to as our agricultural biotechnology pipeline. And so these field facilities, we have approximately 20 acres, 25 acres in our eastern part of the state, and a separate one, about 10 acres in the western part of the state. And these are dedicated acreage with dedicated equipment that's solely used for regulated items. We have separate storage facilities, and we're certified under the USDA's Biotechnology Quality Management System. And this is a full service capacity, so if somebody wanted to test field evaluations of any regulated material, we do this as a service We cover all the aspects needed for the federal guidelines to do this. It's just, you just got to pay by an acre. And so what this infrastructure allows us to do, at least in the public sector, is get technology. I refer to this as the pipeline from the laboratory to marketplace. It allows us to get a technology to early phase 3 development where we reduced it to practice, we captured some IP, and then we let the big dogs take care of all this regulatory hurdles. And this is exemplified by this. This is a picture taken in 1999. It's a soybean plant carrying the single monooxygenase gene from a Pseudomonas multifida, and it's sprayed with 1 pound per acre dicamba, and this is 5 pounds per acre dicamba. All right, that's in 1999. We started doing field trials in early 2000. This is sprayed with 1 pound per acre, pre and post at V3. See, there's the controls. They're growing, and here we are in 2018, and this is called Roundup Ready Extend. And so the example here is this is what the value of such infrastructure to. And the other example for the students is, remember, innovations start in the public sector. We create those little nuggets of coal, and it's the private sector that takes it to a diamond and delivers it to the marketplace. That's capitalism, right? So, what is all this, genetic engineering, genome editing, what is it all about? You guys know this is our small grains breeder, Steve Benzinger, our soybean breeder, George Graff. And what do all breeders need? Genetic variation. And that's all we're doing. We're adding a little bit of genetic variation into the germplasm that these breeders work with that's not available in their current germplasm, all right? Now, breeders are tapping all the endogenous genetic variation, but they're restricted to with that breeding pool. With these tools of biotechnology, synthetic biology, now genetic variation is unlimited. We could tap it unlimited. The only hurdle is unnecessary regulations, right? And so a person like me in a small state like Nebraska, we can explore things. Here's what we're looking at our sorghum program, our input traits. We're looking at improvement in nitrogen use efficiency, trying to enhance, even though it's a C4, photosynthetic capacity, looking at stomate deposition differences. We now have a sorghum which we reduce stomate numbers by 30%. And now we're evaluating this. How does this impact water use efficiency, right? Very challenging to do something like that with conventional breeding without having these other tools. We're looking at architectural changes, cold tolerance, and then changes in root exudates and how that would influence what colonizes the rhizosphere and how will that impact mineral use efficiency, water use efficiency. On the output traits, trying to look at digestibility in the grains, vegetative lipids. All right, we're trying to see, can we accumulate lipids, especially in the stalks of sweet sorghum, move some of that carbon from sucrose to lipids, and can we use this for the bioeconomy? And looking at modifying lignin applications for various reasons. In our wheat program, very similar traits, looking at nitrogen use efficiency, photosynthetic capacity, virus resistance, sawfly resistance, and this lignin modification, again, that's addressed to sawfly resistance. And the output traits, mostly fructan changes that we're looking at right now. But by far the largest effort in my laboratory is in soybeans, where we have the input traits. We're looking at nematode resistance, various abiotic resistance, aphid resistance, photosynthetic capacity. And what I'm going to talk about today is trying to elevate oleic and stearic acid for baking applications, and looking at these high omega-3s, EPA and DHA, for fish feed applications, and this high-value carotenoid astaxanthin, which gives you the colorate you see in farm-raised salmon and shrimp and so forth. And of course, we have an ongoing effort to try to increase total oil protein, and very challenging given these two are indirectly related, but we're trying to address that. And lastly, we have an ongoing one with transposon tagging. Okay, so I'm currently on a project funded by United Soybean Board that's headed up by Leah McHale at The Ohio State University. And what we're trying to do is raise total oil content without compromising protein, with the goal of trying to get over 23%. Now this is challenging because I just said you got this inverse relationship. And my role in this project is trying to address two aspects. We're trying to do a capture and pull towards oil. And a capture is trying to introduce transgenes to enhance photosynthetic capacity, and then stack those transgenes with the different transgenic alleles that will pull carbon towards oil during reproductive stages. Now, I'm not going to be able to go through all this effort because that's a talk in itself. What we're doing is both in oblate leaf phenotype, which is the vast majority of germplasm soybeans, and we're also looking at these lanceolate leaf genotypes because these genotypes allow more light through the canopy, which influences total oil and protein from top to bottom. And I'm going to show you that word rationale. But what I'm just going to talk to you, some of the pool transgenes that we're introducing is this transcription factor from Arabidopsis wrinkled 1, a couple of DGATs from different organisms, and another Cas2 enzyme from Arabidopsis. And I'm only going to talk about this top one right here, the wrinkled 1 transcription factor. Now, the rationale why I'm just going to talk to you while we're looking at both lanceolate leaf and, as I mentioned, the lobulate leaf, because light through the canopy. And this is an old publication by Harry Krishnan at the University of Missouri, which shows that the top of the canopy, pods at the top tend to have more protein, and as you go down, it tends to have more oil. And if you look at those lanceolate leaf lines, they have other problems. They tend to lodge and stuff, but it's more uniform from top to bottom. You don't see this variation. So we're asking the question, if we introduce those suites of transgenes for capture and pull in both sort of architectural differences, can you capture more protein oil across an acreage basis rather than individual seed basis? So as I mentioned, what we're working with, the first one we introduced is expression of this wrinkled 1, Arabidopsis wrinkled 1 transcription factor, which is a global regulator. It was characterized by Christoph Benning's lab many years ago at Michigan State University. And what we did is we just codon optimized this thing and introduced it into soybeans. And this is the cassette, little cartoon of the cassette. It has the beta-conglycinin promoter, a translational enhancer from tobacco wedge virus, and then the ORF itself. Now, the role when we put this in, we're expecting changes in total oil content, which we did not see. But what we did see is a significant boost in palmitic acid. You can see this is from, I think this is a field trial right here. I'm not sure where this came from. Oh, maybe a greenhouse. But nonetheless, it doesn't. I'll show you the data later. You see, in normal wild-type soybean oil, you get about 10 to 11% palmitate. When you add this transcription factor, you're getting close in the upper teens, sometimes in the low 20s. And then you see your reduction in the downstream fatty acids, namely oleic acid, is where the hit comes in, okay? And so when we take these to the field, you can see, this is a picture of the field, you can see this boost in the palmitic acid, you see the slight reduction in both stearic and oleic acid, and these changes in downstream, okay? And so when you look at this, this is a northern blot, and what we're seeing is upregulation of both ACC and FAT-B. FAT-B is a thioesterase that will increase palmitate and tag. And that's where you see this. Here's the 2 wild type, and these are the transgenic ones. And if you go dissect this out, there's 4 alleles of FAT-B in soybeans, and only one of them are being upregulated. And I'm going to talk about these cassettes later, but I'm just going to show you 1289 does not carry the wrinkled 1 allele, by 1301 does. And what you see, it's this gene call right here, the FAT-B2A, and it's this glycol on chromosome 4. And this is the one that's really being upregulated and acted on by this transcription factor from Arabidopsis that's triggering that elevation in palmitate. So what we decided to do as a sidetrack is we had years ago generated a number of lines in which we introduced the mangosteen sterol ACP thioesterase, characterized many years ago by Calgene, which raises stearic acid. We published that a number of years ago. And then in 2002, we introduced a silencing allele for FAD2-1, a delta 12 desaturase. You block this, you increase oleic acid. So what we decided to do was pull out these old events that we had in the cooler and start doing some crossing. We created stacks with the wrinkled in the mangosteen, wrinkled in FAD2, and the mangosteen and FAD2, and then a triple stack. And this is what the outcome was. Right. Okay? So we had, let me walk you through, all these are under seed-specific promoters, either the beta-conglycinin from soybeans or the phaseolin promoter from common bean. In this case, the silencing allele was generated by hooking the FAD2-1 or from soybeans and terminating with a self-cleaving ribozyme. When you do this, it makes an aberrant RNA, and this then triggers the RNA silencing event. Okay? And so, like I showed you, that this allele, that the wrinkled 1, increases palmitate. This mangosteen thioesterase will increase stearic acid, while silencing FAD2-1 will increase oleic acid. And here's what you see here is that outcome of this. All right? This, as I showed you before, expression of a wrinkled 1 increases a palmitate. Expression of the mangosteen now increases stearate from about 4% to over 17%. The silencing of FAD2-1 increases oleic acid from about 20% to over 70%. And then when you start combining the two, here's the mangosteen with the wrinkled, you have a high solids in the mid-20s of palmitate and stearate, but it compromises oleic acid, and you still have these polyunsaturates. When you combine the FAD2 with the mangosteen, you then have a reduction. See this reduction right here? It goes from palmitate from about 10% down to 5%, but then you have an increase in stearate and an increase in oleate, right? Now the triple stack, you don't get the levels of palmitate that you have just with wrinkle alone, but it brings palmitate back to almost wild type. So now we have, the triple stack has a wild type levels of palmitate and still an elevated level of stearate and the elevated levels of oleate. This is what we're going after. Our goal is try to get an oleic acid over 50% and solids in the mid-20s, because that's going to get you the attributes, the functionality for a baking application. All right? Right. And so if you just visualize this, that's that triple stack. All right? At room temperature, it's still a semi-solid. And here's some of the attributes. You can see the increase in oxidative stability of this, and it changes in the melting point. So this is good. But we did it by stacking and crossing. So what we wanted to try to do is, can we recapitulate this cross with a single genetic element? And that's what I'm going to show you back when I showed you those slides. Go back in where I showed you those slides. We did that dissected what fat B alleles is upregulated. So this vector, PPTN 1289, carries a genetic element such as this. It's under the control of the phaseolin promoter, and you have this tobacco wedge translational enhancer. And then you have this intron. This intron carries a hairpin for FAD2-1. So when this is expressed, it's spliced out in the nuclear compartment, it silences FAD2-1, and it simultaneously expresses the mangosteen. So with one cassette, you have two outcomes. All right? So we generated this. That should be the recapitulation of the FAD and the mangosteen stack. And then we added that same, in 1301, we came back and added a wrinkle 1 cassette. So this is the triple stack, and that's a double stack. And here's the outcome of this. And this is, I think this is from the field data. You can see the wild type. Here's two of those 1289s that this should be elevated in stearic and oleate. And then 1301 is the triple stack, where you see a come back, a higher level of palmitate back to almost wild type. And you can see these two, when you just have the stearate and oleate, you see a reduction in palmitate. This brings it back, the wrinkled one will bring it back almost to wild type, right? And that's where you can get the more functionality in solids. You don't want a high palmitate, because then you got like palm oil, right? That's supposed to be bad for your heart, crap like that, right? But this is good, and you have the simultaneous reduction in the polyunsaturates. And that's what you see. That's from a field harvest of last year. That's soy butter. This is good, right? And that's your conventional. You can see this is just basically the same slide I showed you before, but from the field trials. These are plots, right? That's good. Now, you always get the question, do you have impact in germination? And the answer is yes, we do, but it's not that bad. Under like 25C, you know, there you can see some impact. That's wild type. These are 25 seeds. We got like the hit maybe from about 20 seeds will germinate, and then under 10 seeds, so it's a little bit more of a hit, all right? But it's not, like I said, in some cases it's not that bad. Here's that 1289 and 1301 under 25C, and then we get a hit when it's a colder temperature. But it's doable. You may have to, if this is going to translate that, you may have to plant at a higher density or something like that, okay? It's not like you're getting a knockdown of like 5% germination. All right, now I'm going to change to a different vignette. Aquaculture, farm-raised fish, right? Now, it's kind of odd that in the United States, we don't permit offshore aquaculture facilities. All the farm-raised fish in the United States is onshore. The vast majority of aquaculture production is from China, right? And it's the irony in the United States is we'll permit oil well in the Gulf of Mexico, but we won't permit a cage to grow fish because that will pollute the water. Now that's funny, isn't it? So, where does most of our soybean go? Chickens and pigs. And only this little slice, about 1% in aquaculture, right? But soybeans is the perfect feedstock. High quality protein, and you can manipulate the oil. And it's already been used for farming. Now we just want to farm fish. Right? So this is a publication from World Resource Institute showing that, you know, the wild fisheries are still meeting demand pretty well, and they're maintained well. But as we go on, it's going to be challenging to meet the demand. We're going to have more aquaculture, farm-raised fish. But you have to do it in a sustainable fashion. When people talk about sustainability, they usually think about society, they usually think about the environment. But you also have to think about the economy of it. If it's not economically stable, it's not going to last. These are the trends that are predicted for consumption of food. We're going to generally see less meat, less pork, fish was trying to stabilize, and more chickens. So there's room in soybeans to meet this demand. So our goal is try to get fish to eat more soybeans and develop an aqua feed that's devoid of fish products, right? No fish meal, no fish oil. So a number of years ago, this is just a simplified version of fatty acid biosynthesis, the no palmitate there. A number of years ago, we introduced into soybeans a delta-6 desaturase from a herb called borage. You do this, you make this omega-6 fatty acid called gamma-linolenic acid. And we also introduced a delta-15 desaturase from Arabidopsis. You do this, you basically get a soybean oil that looks like flaxseed oil. It'll get about 50-55% alpha-linolenic acid. You stack the two together, and you get this omega-3 fatty acid, an 18,4 molecule called stearidonic acid. And stearidonic acid has been shown, if humans consume it, can convert to EPA and DHA about 4 times more efficiently than the major land-based omega-3 fatty acid, alpha-linolenic acid. So we wanted to try, just showing you the outcome. When you stack these two together, this is under field conditions, it was published a number of years ago, you get the stearidonic acid at about just in the mid-30s, and you get the alpha-linolenic in the mid-20s. So together you have an omega-3 oil that's over 60%. Now this is very oxidative unstable. The first time we did this, we extruded it, it was oxidizing like that. So as it's being extruded, you got to add some antioxidants in there. That was a lesson I learned quickly. But nonetheless, Monsanto developed something like this. They called it Soy Mega a number of years ago, but they used fungal genes from Fusarium. Same activities, just a different source. So we wanted to test this in aquaculture to see, can this be used as a displacement for fish oil? And so we made a diet formulated with like this, where it was like over 70% soybean protein concentrate, and then you could see the laundry list. And how we came up with this formulation is we took the commercial feed, in this case where we started first with steelhead trout, we took the commercial feed diet, it's called Silver Cup. We sent it off for amino acid profile and proximate analysis, and I sent that information to one of these feed formulator type people. And within 3 days, he gave me this recipe. I thought he was full of crap, but it was like he matched it to the T. It was unbelievable. If this matched the amino acid profile of Silver Cup, just like he did, add this, I couldn't believe it. Anyway, we took this pellet, and we either coated the pellet with this omega-3 enriched soybean oil, this standard soybean oil, okay? And then we did the commercial diet, and we worked with a company called Blue Valley Aquaculture in Sutton, Nebraska. And the outcome was like this, okay? What you see here is the fatty acid profile of the flesh of these steelhead trout. And this is the Silver Cup. That's the enriched stearidonic oil, and that's soybean oil. Now, this is not significantly different than this, and this is not significantly different than this, but this is significantly different than this. And at the end of the day, if you go across here, what you'll see is there's more total omega-3 fatty acids in this fillets that were fed this soybean-enriched, this pellet coated with the stearidonic acid oil. Okay? Okay, I think I'm missing a few slides there, but that's all right. Now, our goal, though, trout, they're like the beagle of the fish world. They'll eat anything, right? But our goal is to try to go into these high-end fin fish. And we're working with a company in Hawaii, which is the only, to my knowledge, the only offshore aquaculture facility permitted in the United States. They also have a campus in La Paz, Mexico, right? And this is, if you guys eat sushi, this is hamachi is the Japanese yellowtail. This is the Hawaiian yellowtail. A very high-end sashimi, right? Very high quality. Now, in any aquaculture facility, the feed cost is over 50% of the operating cost. So you get a reduction in that, that's a big hit on the profit margins, okay? And so in Hawaii, if you ever visit the big island of Hawaii on the Kona side, they have these large cages there. They have about 9 of them. And that's where the beauty about Kampachi, you go from eggs to fingerlings, they can go through the whole cycle. It's not like tuna where they bring them in and fatten them up and then sell them for big prices, right? So they have these large cages where they grow these things at. So you got to generate a lot of feed, so it's expensive, right? So when we went in and tried to use that same formulation we did with trout in this Kampachi, and this trial was done like 2008, that's 2007, 2008. Now, I don't have the control, which was, in this case, it was a feed called skreddering, which this would have been the smallest fish, right? Most of them were dying, looking like this, right? Houston, we had a problem. But nonetheless, I was able to go in and sample some of the fillets from these larger fish to ask the question, can this fish metabolize stearidonic acid to EPA and DHA? And the answer is no. What happens is you get a significant reduction in DHA and EPA, and you get this ETA, which is an elongation of stearidonic acid. So this fish is missing a delta-5 desaturase. So we had two problems here. This fish could not metabolize stearidonic acid, and two, the fish were dying, right? So here's the outcome. There's a publication came from a Japanese group that showed that taurine is an essential nutrient for the Japanese yellowtail, right? Now you guys know what taurine is a sulfur amino acid. It's not incorporated in any proteins, but it's all supplemented in all your grad students. You drink Red Bull and vodka now, right? It's all in Red Bull, and it's all in these energy drinks, all right? Well, fish need them. If anybody has cats, domesticated cats require. So if you feed cats dry pet food, look on the back, it's supplemented with taurine. Without taurine, fish and cats, and even humans, will get a disease called green liver disease, and they die. So what we did is mirrored what these folks did in the Japanese yellowtail with Kampachi. And we formulated a diet that either had 40% soybean protein concentrate or 50% soybean protein concentrate with and without a 5% taurine supplementation. And bang. That's the commercial diet. That's a 40% soybean protein concentrate with 5% taurine. That's without. Now the 50%, which is almost a marine-free product, we're still missing some ingredients, but there was a benefit with taurine. But this is not good, this is good. So now our formulation that we've been focusing on is this 40% soybean protein concentrate. In this case, we coated it with all fish oil. Now we asked a question, we knew stearidonic acid could not be elongated by this fish. We did a blend with 50% enriched stearidonic acid and 50% fish oil. And the outcome is this. That's 78 days or full growth at 278 days. That's a mature fish. You can see DHA, you get a small compromise in EPA, but you have an increase in stearidonic acid, increase in alpha-linolenic acid, and more total omega-3s like we saw in the trout. All right? This is pretty good. Now, it's got to taste good, right? If it tastes like natto, you may have a problem. So we sent some of these fillets off for a taste testing panel. And this is what was fed to these consumers. Now, you can argue if you put enough wasabi on something, that you're done. But the consumers, this is just like just showing this whole massive data. They could not differentiate by looks, smell, taste, the difference between what I call our Nebraska soybean diet versus this commercial diet. This is pretty good. Right? So we communicated these outcomes in this paper here. And then there was another group in Nebraska that did the economic feasibility, assuming what the cost of stearidonic acid, if it ever got onto the market, would be. And it matched. It's pretty good, right? So the economics were there. But this is just like a paper demonstration. What we also did, can we further displace fish oil in this 40% soybean protein concentrate taurine formulation? The answer is no. The 50-50, you see this, but the more you try to displace fish oil with the stearidonic oil by 75-25 or 90-10, you're starting to take a hit. So we couldn't do that. But we asked, can we displace more taurine? Because taurine goes for about $4 to $10 a kilo. And so we tested the 5% taurine, 3% taurine, 1% taurine, 1% worked fine. So now our formulation is 40% soybean protein concentrate, 50% stearidonic oil, 50% fish oil, 1% taurine. Trying to get the cost down. Now, remember I told you our goal is to try to get a marine-free product. We then went in and tried to design a formulation comparing it to this 40% soybean protein concentrate supplemented with 1% taurine, the 50-50 blend, but now going to a marine-free product and adding an inclusion of an algal paste. In this case, we did a mixture of Tetracellus and Isocristes, and the reason we did that is because these organisms have EPA and DHA, and also Tetracellus produces endogenous taurine levels, right? Now, we did this. This is almost a completely marine-free product. There's a little animal stuff here, blood meal, that comes from all kind of gross stuff, but you put it in there, all right? We did a feeding trial comparing these two. This works, right? We can get, with this algal inclusion, we can get, I won't show you the growth rates, but the growth was comparable. That's the algal paste. You can see the changes, no changes, are relatively same in EPA and DHA. The problem is the cost. That algal paste for 15 kilos of that algal paste costs $12K. It's not going to happen, right? So we're now trying to work on avenues. Can we improve taurine production and growth of targeted algal species to have this inclusion? I'm now going to talk to how we're adding further, trying to develop the soybean to have co-products at additional values, and we're producing soybeans with tocotrienol. And the reason we're putting tocotrienol in there, it's a form of vitamin E. You ask the question, if you feed that to the fish and you get high levels of tocotrienol in the flesh, will that increase the shelf life of that fillet in the market? Okay? And so what we then did, right now we're at stearidonic acid right there. That's 2 genes. We added 2 more. We added a delta 6 elongase and a delta 5 desaturase to try to get EPA. And we wanted to make that high-value carotenoid called astaxanthin. And this astaxanthin in something like farm-raised salmon is about 17% of the feed cost. It goes between $500 and $1,000 a kilo, right? You're kind of approaching cocaine levels, right? So what we first did is we put in a triple stack from marine bacterium and one from maize, phytoene synthase, and the CRTW and the CRT, where's the other one? Oh, CRTZ right here, all right, to get astaxanthin. That's 3 genes. So we have 4 genes for EPA, 3 genes for astaxanthin, 1 gene for tocotrienol. Worst we did is did them all independently. We put in the 3 gene stack for astaxanthin and additional 2 genes for EPA. What you're seeing here is this EPA additional 2 genes by himself. Now what you see is only you don't see any of the desaturase, you just see changes in the elongation. All right, so you get these 20 chain molecules coming up. All right, so what we did is we take these events and we crossed them with our stearidonic acid line. And then we introduced the lines that we're trying to get astaxanthin, and we just selected for yellow color. And these, when you do this is the percentage of the carotenoids in each one of these beans. And the vast majority of it was beta carotene. But we did get some amounts of astaxanthin. And we brought all those together by crossing. And what you're seeing is, just for this alone, just making EPA, we can get about maybe 3 to 4%. Fish oil has about maybe 5 to 9%. DHA is a little bit higher, so that's our goal, is try to raise it a little bit. But we're getting astaxanthin about 25 micrograms per gram seed, and at that level, you'll need about 4 million acres of soybeans to satisfy what's required of the annual supply of astaxanthin, right? But when we bring these all together and try to bring them together, we saw astaxanthin maintained itself, but we saw a reduction in the EPA levels. But nonetheless, with that 7 gene stack, that's what that oil looks like, nice red color, right? This is kind of a prototype where we want to go. So what we then did, we used these tools of synthetic biology to order all these genes and paste them together with golden braid. And we changed the carotenoid pathway. In this case, we took a little hint off of this plant cell paper. The initial pathway was from this marine bacterium. Now we're using this, taking advantage of what these folks found out with this Adonis genes, and we put in these 3 genes in the soybeans. And what this does, when you do this Adonis pathway and not the bacterial pathway, there's the first one, you see this high level of beta carotene. With the Adonis genes, we don't see that, only see the astaxanthin, and this is what we want, all right? So then, taking advantage of this information, we had this both in camelina and in soybeans. What we then did is stitch them all together in one big vector, right? We have the suite of genes for tocotrienol, astaxanthin, and EPA. One shot. This is what I like. Genome editing's good, but man, when you can start putting pathways, that's where the power comes in. And bang. Nice color astaxanthin. I won't show you the data, but the tocotrienol levels through the roof, and we have EPA close to 9%. This is good. What's the problem? Oh, that's just from the field. Last year, showing the astaxanthin levels. The problem's here. Under the greenhouse, you may not see, you can see these are segregated, you can see the red and so forth. But in the field, we're getting a lot of wrinkled phenotypes, right? And we believe this wrinkled phenotype is coming from the astaxanthin. We know it's not the EPA because we can make that alone. This leads to abscisic ABA. And we're thinking there's premature levels accumulating and to get maturity of the seed too much. So we're going back in now, we're designing these vectors for transformation. We're looking at just EPA and DHA and tocotrienol. We're going to do astaxanthin, tocotrienol, testing promoters for early expression and late expression. My guess is on the late side. Ed Cahoon thinks it's on the early side. But we're going to test both to try to alleviate this wrinkled aspect. And then we're going through the game plan based on what we learned from this for what we think could be the true feedstock for aquaculture, where you have these co-products, the tocotrienol looking at stability of the oxidative stability of the fillet and increase the shelf life of the product. These two to displace fish oil and that high-end carotenoid for other applications. And you still had the high-value protein, right? Okay. Now for my commentary. I stole a quote from Hunter S. Thompson, when things get weird, the word term pro, because I was watching the news last week with my dog Murphy. I was sipping scotch and he was with his favorite bone, and this came up to my mind for some way. This quote came to mind. Now, here's where it is. Here's what bothers me. We got people talking about we regulate the process, right? The regulations of all these biotechnology stuff. We regulate the process by which we do things, right? Whether it's these genome editing tools, you still need to introduce these reagents, either direct DNA delivery or some sort of agrobacterium based, right? And these mutagenesis, EMS or fast neutrons, they're not regulated. This is still in question, but may not be regulated. Right? Now, why is that a problem that the world regulatory agencies regulate the process and not the trait? It's exemplified here with hyoleic acid. Right? Vista Gold, that's Monsanto's hyoleic acid. Plenish, that's DuPont's, or what's your new name now? Arta Gold or something? Cativa? That's Pioneer's hyoleic acid. These are done by silencing. The only difference, this silence is only FAD2-1. That's FAD2-1 stacked with the silence of FAD-B and stacked with the silence of FAD3. Got a little bit higher oleic acid. But a company called Calyx that's based somewhere around here, they did it by genome editing. And folks at USDA in Missouri did it by a mutagenesis approach and selection of endogenous alleles. Not regulated, likely not regulated, regulated, but the trait's the same. Why? Why would we say that this needs regulation, but these don't? The outcome's the same. If they regulate the trait and not the process by which they make that trait, all good. What's the second reason that's a problem, that they regulate the process and not the trait? There's no competition for generics. Exemplified here, Roundup Ready's off patent. I can go into my laboratory and generate 1,000 Roundup Ready events in soybeans. But each time I deliver that transgenic allele, the same one that's been on the market since 1996, I'd have to go through the regulatory process. That's the problem. If I would be able to do this, I'd be able to hand off a generic version to our soybean breeder, George Greff, at a fraction of the cost. We don't have the luxury that we do in ag biotechnology that they do in the pharmacology business, right? Once a drug comes off patent, competition comes in, bang, they can deliver it, and you have it at a fraction of cost. We don't have that opportunity in the ag biotech world. But you'll hear the argument, well, you know, the consumer wants regulation because that gives them confidence. Well, not really. They really don't. And here's the example, is the supplement industry. You have a problem with your joints, your sex life bad, I got a pill for you. and you could take it without question. And that's a multi-billion-dollar industry, no regulations. So this argument that regulations gives more confidence, not true. This is my laboratory. These are the people that helped me with the feeding trials and so forth. Shen did some of the dissecting of the wrinkled one. Ha-Yoon Park was a former grad student. He did most of the work for both of those projects I described. Hey Jin is our person in UNL that's stitching things together for Golden Braid. This is my laboratory. Han and Lily do our soybean transformations. The rest look up to me as if I'm God. These are the funding agencies that support my laboratory, and I want everybody to remember that the ability of the United States of America to produce a plentiful and safe food supply resides in the American farmer. And again, I thank you for the invitation.
Thank you so much, Dr. Clemente, for your presentation. Are there any questions?
Yes.
Floating mics too, so if you could use a mic, that would be great.
Thank you for your talk. That was excellent. This whole project, was it designed with a specific species of fish, or can you give this feed to any type of fish, or do you just change the formulation?
You'd have to, the oil would change the same, the protein, the ratio of, first a level of oil. So this fish, this Kampachi, needs a high level of oil. It's like 19%. Different fish need different oil levels, but the fatty acid would change the same. And then the mix of the protein levels, the amino acid, how much soybean protein, that would change. That changes by species. So the United Soybean Board has trials going all over the world to try to displace fish meal in all sorts of avenues. They've been very successful at that, but they ignored the fish oil component. So we're the only one that they're funding that's looking at the oil. But the only change is what's the ratio of amino acids, i.e. level of soybean protein concentrate, or if you just want to use a straight meal, that's the change that would have to be. But the same oil could be used across the board.
Other questions?
So you're operating within a space of farm-raised fish in a market where wild caught are often valued. You're also talking about transgenic product, and I was wondering if you see those as any barriers to market adoption, or if your idea for this is that it might be more popular in other foreign markets rather than a very health-focused right now America in terms of sushi.
Right, so my vision is to say, this is where the technology is at. I didn't develop any of this technology. I just put it all together what other people did. And what I'm arguing is we have to educate, like Rex was mentioning yesterday, we have to do a better job as a science community to educating the non-science population of what this technology is, what is this not. So when you hear these terms, non-GMO, organic, that's a marketing scheme. It has nothing to do with health attributes. It has nothing to do with environmental benefits. And this is what we're as a scientific community, we're not doing well on. And like I said, if you had somebody up here who's going to be developing a oleic acid soybean, what they boast about it when it's EMS or the genome editing, what they're going to science, they're going to say it's non-GMO. What is the consumer here? I knew there was something wrong with that thing called GMO. And it's all our fault. And I believe what I'm going for is this is a demonstration. There's not much I can do alone. But as a community, we have to do a better job to educate the non-life science community, again, of what their food supply is. What I would argue is that the way we grow our food and the technology we do to grow our food, not a problem. It's what we do post-harvest. It's all you food scientist people screwing things up, right? And what we're also not doing well is people aren't cooking, right? They'll go to McDonald's because you can get 4 hamburgers for $5, right? You can't beat that. But if you learn, we have to go back, you can take it, you can get a whole bunch of raw meat, cook it yourself, and you can last all week. That goes a long way. We're not doing a good job in that. So I think this sort of stuff that is probably going to sit in the shelf until I'm dead and gone can get off the shelf all over the globe if we do a better job in educating society of what this technology is and importantly what it's not. And the folks like at Whole Foods, they're making a living off of GMOs because they're the scare tactics. The whole organic community is pushing this stuff to scare the non-scientific community, right? Now, if you asked any scientist, you'd probably get a consensus of over 85% that the process to how we deliver this genetic variation is safe. You'll never get 100% of anything, right? But if you get over 85% of a scientific community agreeing on something, that's pretty damn good. So to answer your question is, my goal is to demonstrate it and little by little do my part to try to educate the population about this technology. Now, sometimes if you get me going, I get a couple drinks in me.
All right, that's all the time we have for questions.
Oh, yeah.
Let's thank Dr. Clemente
you.
once again. Now I'm going to turn it over to Emmanuel to introduce our next speaker.
Hello, everyone. I am very pleased to introduce our next speaker, Dr. Natalia Dideriva. She is a distinguished professor of biochemistry at Purdue University, Indiana, and her lab studies metabolic pathways and how plants produce SANs and different metabolites. Our talk today is titled Plant Volatiles, How Plants Synthesize and Emit SANs and emit them. Without further ado, I would like to invite Natalia upstage for a talk. Please, let's welcome Natalia.
Thank you very much for your nice introduction. It's a great pleasure to be here, and especially big honor to be invited by young people and see a lot of young people in this audience. And before I did not forget, I will tell that I have open postdoctoral position in my lab, because I always forget to say it at the end of my talk. And if somebody is interested, just talk to me. And today I'm going to talk about plant volatile compounds. And this is small molecular weight lipophilic molecules which have a low boiling point and high vapor pressure at ambient temperature. And we are living in the world of volatile compounds. They're actually all around us. They're heavily used in perfume, cosmetic, and fragrance industries. Also, aromatic compounds, biologically active aromatic compounds, and essential oils, they use in aromatherapy, actively used in late years. And humans realize about importance of volatile compounds in their life for a long time. And even in 3000 BC, Egyptians already were making these primitive perfumes and use them for religious ritual. The importance of volatile compounds for our life is also highlighted by the fact that in 2004, the Nobel Prize was awarded to Richard Axel and Linda Bach for discovery of odor receptor in humans. And they discovered this huge family of receptors and showed that practically when we inhale volatiles, they get into our nasal epithelium receptors where they localize receptors and they transfer the signal through nerve system to our brain. And then we have a memory about some kind of scent, and we have memory from our childhood about good and bad scent. But the majority of volatile compounds practically produced by plants, and they release from all plant organs, from flowers, fruits, vegetative parts into the atmosphere, and from roots into the soil. And the primary function of these volatile compounds in plants are to defend them against herbivore and pathogen, and to provide a reproductive advantage by attracting pollinators and seed dispersers. And this is how we can use probably this research to make a biologically pest management strategy, just to use natural plant defense system to protect plants against herbivore. And volatiles emitted from as a part of plant defense, they can directly intoxicate or repel attacking herbivore, or they can attract natural enemies of attacking herbivores via triotrophic interactions. And by emitting these volatile compounds, plants not only reduce amount of attacking herbivores by 90%, but they also inform neighboring plants about pathogen attack. Volatiles released from the roots contribute to below ground defense system, and they also serve as repellents or antimicrobial substances, and they also can attract natural enemies of root destroying herbivores. Volatiles released from plants, they provide chemical cues for pollinators. And in general, flower shape, size, and color. This is important characteristics of flower which attract pollinators to flower. However, if flowers can be identical in color, shape, and size, there are no two scents which are exactly the same due to high diversity of volatile compounds and their relative ratio within the scent bouquet. Volatiles emitted from fruits, they determine taste and flavor quality of fruits, and they also attract seed disseminators. Considering importance of volatile compounds for one more point. Today, about 1,700 different compounds have been isolated from 90 plant families. And they constitute about 2% of secondary metabolites produced by plants. And these volatile compounds are presented by terpenoids, monoterpenes and sesquiterpenes, phenylpropanoid and benzenoid compounds, fatty acid derivatives, and amino acid derivatives. Volatile compounds also contribute to atmospheric chemistry because they produce these secondary aerosols, which served as a cloud concentration nuclei. And in such way, they contribute to atmospheric chemistry and climate. And benzoate compounds alone have been estimated to emit about 10 to the 9th carbon per year. It's only benzonates, which are not the major compounds. The major compound will be isoprene, of course, and terpenoids. And considering importance of volatile compounds for humans, plants, and atmospheric chemistry, we ask several questions. How plants make these volatile compounds, and what are the metabolic pathways, enzymes, and genes involved in scent formation? How is emission of volatile compounds is regulated? What are the molecular mechanisms that control volatile emission during flower development, a daily light dark cycle, and after pollination? Where these volatile compounds are produced in the cell, in flower organs, in what kind of cells, and where inside the cell? And could we change volatile profile or improve volatile profile? And what will be effect of this targeted metabolic engineering on steady state level of volatiles and fluxes within pathways, on insect perception, and also how we can improve, for example, quality of our flowers, since during breeding, all breeding processes were focused on color, shape of flowers, and post-harvest life. And practically, since there is negative correlation between post-harvest life and scent production, we bred out scent from cut flowers. And to answer these questions, initially we used two model systems, bee-pollinated snapdragon flowers and moth-pollinated petunia flowers. And Snapdragon flowers produce scent during day, and a scent profile consists from terpenoid compounds shown here in yellow and phenylpropanoid compounds, while petunia flowers produce predominantly phenylpropanoid compounds. When we started this work 20 years ago in my lab, no genes responsible for formation of these compounds have been known. And even today, only genes responsible for 5% of volatile compounds have been identified and characterized. Today, we practically know all genes responsible for formation of all these compounds. And also using this system, two systems allowed us to compare formation of these compounds, the same compound in diurnally and nocturnally emitted flowers. And what did we learn here? We learned that formation of a floral scent bouquet includes the coordinate expression of genes encoding enzymes involved in scent formation, in the final steps of scent formation. And you can see here that, for example, in petunia flowers, scent compounds are produced in this corolla where the highest expression of different genes are found. And here is the gene responsible for benzyl benzoate and ethyl benzoate formation, and another gene for methyl benzoate formation. And in snapdragon flowers, formation of these volatile compounds occurs in upper and lower petal lobes. And here you have genes which are coming from different pathways. For example, this is benzenate compound, BMT, responsible for methyl benzoate, and this is two genes responsible for benzoate terpenoid compound. And they're also expressed in the same part of the flower. And over flower development, you can see this coordinate expression of genes, and you always see the peak of expression about 1 to 2 days before peak of scent emission. What we also learned that the level of substrate in a cell is involved in regulation of developmental and rhythmic volatile emission. And if you look on this is developmental emission profile in snapdragon flowers, and you can see that activity of gene, BMT gene, is going up and down. But old flowers have similar enzyme activity as young flowers. It means that they capable produce the same level of methyl benzoate, but they produce much 3 times lower. And when we check the level of substrate, which is benzoic acid in this particular case, you can see that the level of substrate exactly 3 to 5 times lower in old flowers than in young flowers. Also, if you look on rhythmic emission, for example, you have rhythmicity in emission, and this is again snapdragon flowers, you can see emission during the day, but activity does not show any oscillation. It means that during night, these flowers are capable to produce a lot of methyl benzoate. However, they don't produce because practically you don't have substrate there. The level of substrate also determines the type of product it's produced. For example, in snapdragon flowers, gene which is protein which is responsible for formation of methyl benzoate is benzoic acid carboxymethyltransferase. And it's used only benzoic acid and acetoacetyl to make methyl benzoate. In petunia flowers, it's benzoic acid salicylic acid carboxymethyltransferase. What it means that it can use benzoic acid and salicylic acid and produce methyl benzoate and methyl salicylate. Moreover, biochemical characterization showed that enzyme is catalytically almost 100-fold more efficient with salicylic acid than with benzoic acid. However, petunia flowers, they don't produce methyl salicylate because the level of substrate, which is salicylic acid, is very low. In contrast to petunia flowers, Snapdragon flowers have salicylic acid carboxymethyltransferase, which can also use benzoic acid in addition to salicylic acid as a substrate. However, this gene does not contribute not to methyl benzoate formation and also does not produce methyl salicylate because expression of this gene is very low. Now, knowledge about substrate is very important for metabolic engineering as well. And here it's a very good example. When we overexpress rose alcohol acetyltransferase in petunia flowers. And this particular enzyme can produce geranyl acetate from geraniol. And when we overexpress it in petunia flowers, we thought that we will produce geranyl acetate. But instead, we did not see any geranyl acetate, but we saw huge increase in benzyl acetate and also some production of phenylethyl acetate. Then we went back and biochemically characterized this enzyme because initially we used only geraniol as a substrate. And what we found that indeed it can use benzyl alcohol and ethyl ethanol as a substrate, although of course catalytic efficiency was significantly lower. Since these substrates were available in petunia flowers, in cells of petunia flowers, this is what this introduced enzyme uses. But when we fed petunia flowers and leaves with geraniol, we started to produce geranyl acetate. This is why for metabolic engineering, it's very crucial to know if your substrate is available or not. And the function of introduced gene in plant will depend on substrate availability. Now, volatile emission of compounds depends on rate of biosynthesis and rate of release. And for many years, for all these years, it was assumed that volatiles is passively diffused out of the cell. And I will show you that this is not the case. Let's start with rate of biosynthesis. And as I mentioned already, it's species-specific, spatially, developmentally, and diurnally regulated. And closely related species, which rely on different pollinators, they produce different volatile compounds. Overdevelopment, emission will be highest when plants are ready for pollination. And also, during a daily light cycle, very often emission correlates with activity of potential pollinators. And here you can see, for snapdragon, I said it's depollinated during the day, and petunia is most pollinated during night. And our rate of biosynthesis also depends on physiological factors, such as pollination in floral tissues and pathogen infection in vegetative tissues. And after pollination, flowers practically decrease scent to direct pollinators to unpollinated flowers. However, it takes about 48 hours in snapdragon flowers and 36 in petunia before they shut down scent production. And this is exactly time how long it takes for pollen to move through the style and get to fertilize the egg. It means that fertilization triggers shut down the scent, and flowers, they need to be ensured that it's pollinated. Rate of release. In order to volatiles to get out of the cell, they need to cross plasma membrane, cell wall, cuticle, and then they get to the atmosphere. In this case, rate of release depends on physicochemical parameters of compound by itself, its volatility, diffusivity, octanol water partitioning coefficient, and the physicochemical properties of the barrier, like membrane lipid composition, cell wall porosity, cuticle composition, and permeability. And rate of release will also depend on environmental factors such as light intensity, temperature, and relative humidity. And we ask the question, are biological processes such as sequestration, transport, or shuttling mechanism involved in release of volatiles out of the cell? Just to get idea where these volatiles are made. In non-green organs like flowers, volatiles are produced in these chemical cells in an inner epidermal cell layer and a little bit less to outer epidermal cell layer. And within the cell, this is conical cell, you can see that methyl benzoate is produced here in cytosol because it's a huge vacuole. Here it's pushing cytosol against cell wall. And for example, terpenoid compounds they produce in plastids. You can see here. In non-vegetative tissue, these volatile compounds often accumulate in trichomes, and you can see here from basil and rosemary. When they are not accumulates in trichomes, they produce not in epidermal cell layer. They produce in palisade mesophyll, where, for example, it was found isoprene synthase localized. And although they can get out through somatic here, they still need to cross cytosol, plasma membrane, and cell wall barriers in order to be released out of the leaf. As I mentioned already, plant volatiles are typically low molecular weight, lipophilic, and they have high vapor pressure. And for them, it will be very favorable to stay in membranes. But it will be very unfavorable to diffuse out of membranes to the cell wall, which is active. And diffusion model, in addition, it lacks membrane selectivity directionality and also control over toxic effects of some volatile compounds. Also, what we saw over all these years, that for example, internal poles not always correlate with emission of volatile compounds. And here you have example for methyl benzoate. And you can see here, that for example, we have emission in young flowers, but it's no accumulation inside cells. And this is really very easy to imagine. You synthesize and emit right away. However, in old flowers, you do see huge accumulation inside cells without emission. And if it's simple diffusion, what keeps these volatile compounds inside cells? And then what we decided to do, we decided just to make a calculation. Since we know how much is emits, how much is accumulate, what concentration of volatiles should we have inside the cell if it's simple diffusion process? And we use a fixed first law where you can see concentration along, emission rate will depend on concentration, difference along the path, it's every plasma membrane, cell wall, cuticle, and resistance to diffusion across. And this resistance depends on the length of the barrier and properties of volatile compounds. And at steady state mass balance, at each barrier, we should have rate of biosynthesis at the end equal to emission rate. And what did we get? What we found is that it should be 100 millimolar in plasma membrane for methyl benzoate and 48 millimolar for terpenoid compounds mirrolidol. This is really toxic compounds levels. They will completely destroy the cell. They can get to leakage of cytosol out of the cell, and it clearly suggests that biological mechanisms must exist to lower barrier resistance. And this is why we started to look how these volatile compounds are getting out of the cells. First, we look what potential mechanisms could be involved. And here we saw that these volatile compounds could be carried out to plasma membrane in vesicles bilayer or lipid transport proteins, and that at the level of plasma membrane, probably transporters can take it across plasma membrane. Likely, LTPs, lipid transport proteins, can carry it out cell wall, because otherwise they will go back to plasma membrane, and from cuticle it will be simple diffusion to the atmosphere. And first we decided to search for transporters across the plasma membrane. And what we did, we generated RNA-seq data sets at a stage with the lowest emission and stage of the highest emission. And our hypothesis was that biological players involved in VOC emission will be transcriptionally regulated with phenylalanine level, which is precursor for all these phenylpropanoid compounds in petunia flowers, and volatile bisynthetic genes. And here we check all genes that our RNA-seq data sets is correct. We check expression of all genes involved in Phe biosynthesis and downstream for phenylacetaldehyde formation here, and spouse the first step. And we did a search, T-BLAST-N search for with ABCG Arabidopsis wax transporter with known function, considering that our volatiles have similar, probably physicochemical properties as a wax. And we found several candidates which were upregulated on day 2 versus the day -1, and also were upregulated in the evening versus the day. And one candidate showed 52% identity, 72% homology to the wax transporter from Arabidopsis, and it was upregulated 100-fold. And when we look in the literature, we found that this is ABCG1 transporter with unknown function, but it's localized already to plasma membrane, which was very helpful for us, and in addition was upregulated by ADARANT1 transcription factor, which upregulates these pathways involved in volatile formation in petunia flowers. We thought it's a very good candidate, right? We downregulated it. We generated 3 transgenic lines with downregulation more than 75%. Total emission went down right away, internal pools accumulates. And we had practically effect on all volatile compounds. Here it's emission, this is internal pools. We overexpress this transporter in tobacco BY2 cells to prove biochemically that it indeed can take volatile compounds. And we did our assays with radiolabeled methyl benzoate. The way how we did, we took cells, we added methyl benzoate radioactive, and then we measured activity. And you can see if, for example, this transporter can take this compound out, we should see some decrease in labeling in cells. Indeed, we found that cells overexpressing this transporter had less methyl benzoate inside. We also checked substrate specificity, and you can see here that it can take methyl benzoate and benzyl alcohol, but it was not able practically take any terpenoid compounds like menthol, which is monoterpene, and this is diterpene analog. What is about our prediction about toxicity of these compounds? Because always when we talk about toxicity and in the literature results, they always showed by fitting experiment, which was not physiologically natural. You just fit it. And what we decided to do, we decided to take 2 different dyes with different mode of action to see if really these compounds are toxic for cells. And we fed, we stain, not fed, we stain cells with we stain flowers with propidium iodide, which would only stain nuclei if a plasma membrane is damaged. And you can see here why you don't see nuclei in wild type, you do see it very nicely in transgenic plants. We also use fluorescent diacetate, and this stain, it stains cell viability and membrane integrity. And the way how it works, it can get in the cells, and the dye is not fluorescent until diacetate moiety is removed by esterases. When it gets in the cell, diacetate moiety removed, it becomes fluorescent, but it cannot get out of the cell if it's not damaged. And you can see less in transgenic lines. We also mimic this situation, and we started to feed flowers with benzaldehyde, and you can see we were increasing the level of benzaldehyde, and starting from 50 millimolar, we started already to see some nuclei and 30 millimolar, and this is internal concentration of benzaldehyde. And that's our proposed working model for now, that volatiles would get in bilayer of vesicles. They will move to plasma membrane. They will be released to plasma membrane. Then ABC transporter will take it across plasma membrane, and then lipid transfer protein cross, and then they will volatilize. And actually, this model also shows you new targets for metabolic engineering and for improvement. And in general, metabolic engineering of plants is very important because it can improve scent and aroma quality of flowers and fruits. It will increase value of ornamentals. It will produce novel aromas for perfume industry. They can use as a natural factories for food industry. We can produce spices even in flowers. And we can produce biologically active compounds for aromatherapy. We can increase yield of insect pollinated agricultural plants because they highly depend on pollination and quality of pollination. And quality of fruits depends on amount of ovules which is fertilized. And it also will enhance natural defense of crop plants and provide an alternative pest management strategy that is based on biological control. It will uncover effect of target metabolic engineering on levels of metabolites and fluxes. And also, we still don't know how insects perceive these compounds, and it will provide, will uncover the role of individual volatiles in pollination attractors and plant-insect interactions. And this is very important because sometimes the same type of compounds are attractants and repellents, and how do insects distinguish this? It's still unknown. And I want to thank all people in my lab who participated in this work, and my collaborators, and this is engineers who are doing modeling, and we are working for more than 15 years now. My collaborators in Belgium who did these assays with transporter. We have some kind of metabolic engineering work with Hebrew University of Jerusalem, and some subcellular localization with Max Planck Institute for Chemical Ecology in Vienna, Germany, when I had my sabbatical 15 years ago. And this is fundings, and I really appreciate fundings from National Science Foundation, which I had for all these 20 years, and also from Alexander von Funden Foundation, which allowed me to have sabbatical last year in Germany. Thank you very much, and I will be happy to answer your questions.
Thank you very much, Natalia. We can take questions for Dr. Dudariva.
Thank you, Dr. Dudariva.
This is a fascinating talk for me to hear working in fruit aroma. I noticed on a couple of your slides, linalool was a compound that was mentioned, and that's a really interesting one for us in strawberry because it's produced at high abundance, but we don't have much evidence that it improves aroma or flavor for humans. And I'm just curious, from your knowledge of linalool in many crops, what are the potential functions of linalool, whether in resistance to herbivory or pollinators or anything like that? Just curious what you know about it.
What I want to say is that abundance of compounds, it does not reflect how much we perceive it and how much is the flavor, because for each compound there is a threshold, and sometimes even minor compounds contribute much more to our sense of flavor than highly abundant compounds. Across species, you know, this is one compound which I still cannot fully understand how it's produced. Because if you look and compare with ocimene or myrcene, which is similar type of compound, they can always follow. You increase pool size, they're increasing. Linalool, it's doing something different, and we are still working on it, trying to understand, because we have a mint project, and we sequence 52 different species from mint family, and we are right now looking on linalool, how it's produced. Of course, linalool in citrus fruits will be contributing much more, but to say that it will be the major one, I would not say like this. Because it's really, it's a mixture, and it's always mixture. It's not one compound. And insect in the same way, they don't perceive one compound. They perceive the ratio of these compounds. And we did these assays and studies.
Other questions for Dr. Rivera? Okay, thank you very much. Let's thank her once again.
Thanks, Emmanuel. All right, so next on our schedule is break. So let's thank again our first two speakers this morning. So during break, we have donuts, or bagels in the back, sorry. And we also have lots of coffee to keep you going this morning. We have notebooks in the back left, so you can take some notebooks. They have our nice little logos on them, so make sure you take good notes during the symposium as well. Down the hall is our poster session, and it's in the Cherrywood room. So this would be a great opportunity to check out posters during the break, and we will resume here at 10:15. Do you have anything to add? Nope. All right. See you back at 10:15.
I don't know if I messed this up.
Oh, yeah.
Because I closed her so I could take it out.
Oh,
Did I break it?
that looks good.
Just for a break anyways, so.
I'm gonna grab my coffee now. Is this the bag for that?
Bag? Oh yeah.
Bag.
Break and refreshments, right? Yep.
I thought you said donuts instead of bagel.
Yeah.
I cannot think of the word bagel.
Yeah, we're so excited. And I know, and now we're all crushed.
Now or later.
Whenever he's comfortable. Where is he?
He's talking there with like Spanish money.
He can talk to him during lunch.
Whatever is more comfortable for you and him, because at lunch he was also really eating.
Yeah, we like the idea, but not money.
I couldn't see if you're going to use the clip-on. Can we test it first? Okay. Well, we can just ask. We can just. I mean, like what he said is fine. Like it is perfectly okay for us to just say advance. You know, can we test this first?
None of legs.
They'll be here around.
Oh shoot! Do we have advances?
This isn't still mic'd right to the live feed. It is. No, I mean.
Just an announcement, we're going to get started in about 2 minutes, so if you want to start finding your seat, we'll resume in 2 minutes.
Okay, if everybody could go ahead and find their seats again, we're going to start the next speaker. So Yaya will come up and introduce our next speaker.
Hello everyone. It's my pleasure to introduce our next guest speaker, Dr. Vance Vetager. Dr. Vetager earned his MS and PhD degrees here from University of Minnesota in plant breeding and molecular genetics. Since 2009, Dr. Vetager has directed the strawberry breeding program at the University of Florida, which develops cultivars that are widely grown in Florida and around the world. Dr. Vetager's cultivar development program is enhanced through genetic characterization of traits, primarily disease resistance and fruit quality. His today's talk is on breeding for a complex quality traits in a complex fruit crop. Please join me to welcome Dr. Vetager.
Thank you. Thank you for the introduction, and it is a real pleasure to be invited to speak here today. And as Tabari mentioned, it's a double pleasure because I was, as a student, part of the organizing committee for the very first Pioneer Symposium in 2008, right? This event has really grown, and it's really fantastic to see what it's like today. So thank you for inviting me. Well, today I'm going to talk a little bit about fruit quality in strawberry, which is a very complex challenge, both because I'm working with an octoploid organism and because quality has so many components when it comes to strawberry. As Yaya mentioned, I started breeding strawberries in Florida about 8 years ago, but the breeding program has been going in some form or fashion since 1948. With some open pollinated selections in the early 1950s, and then a continued crossing and selection scheme through the 60s, 70s, and 80s. And I sort of have a pedigree in a couple of senses. You know, plants have pedigrees, but I also have a pedigree, both on the Florida side and the Minnesota side. So Stan Hokanson, who's there in the back, was my advisor for my master's and my PhD here. Many of you may not know this, but Stan, before coming to the University of Minnesota, was a strawberry breeder at the USDA in Beltsville, Maryland. Stan's advisor at Michigan State was Jim Hancock, who's very famous for his work in strawberry. And Jim Hancock's advisor was Roy Springhurst, famous strawberry breeder at the University of California, Davis. So it's really fascinating to see how that kind of got handed down over time. On the top there is kind of the Florida pedigree from the breeding program. Albert Brooks at the USDA in the Plant City area in the 1940s and 50s. Charlie Howard, who is a plant pathologist turned breeder in the 60s and 70s. And then Craig Chandler, who started in the 80s and who is my predecessor. Now, many people may not know this about the University of Florida, but we actually pulled off the first successful human cloning experiment. This is Craig Chandler, who was cloned, and there I am. Actually, I wish I was a clone of Craig Chandler. Craig, as my predecessor, was very, very successful in his job. His varieties came to dominate the Florida industry and in 50 countries around the world. And it's really his efforts that really fund what we're doing today. And one other thing that I'm really grateful for that I want to mention about Craig, and it is a great lesson about plant breeding, is that when Craig retired and the position was opened, which I filled, he arranged it so that he could overlap with 1 year with me, so that I could walk in the field with him every day throughout the season and learn both the science and the considerable art that there is to strawberry breeding. And it was a fantastic experience to pick his brain every day. And it's just something that is a real gift to me, and I highly recommend, if there's any administrators or breeders about to retire in the audience, I highly recommend that kind of arrangement. Well, the Florida strawberry industry is concentrated in the middle of the state around Plant City. So if you've gotten your packages of strawberries in the wintertime, you often see Plant City on the label. And that very geographically dense area has 11,000 acres of strawberries. California is obviously the big strawberry behemoth of North America, but they don't produce a whole lot in the winter, and that's where Florida comes in. And so we're the second leading strawberry producer in the nation. That acreage is made up of basically 3 main varieties from Florida: Radiance, which is Craig released in 2009, Sensation, which Craig and I released in 2013, and then I released Beauty, which is a brand new one just last year. And then about 10% of the acreage is in Triskel proprietary varieties in Florida. But this is kind of what the acreage looks like today in terms of varieties. And these 3 varieties represent in some different ways the types of quality traits that are really important in strawberry in Florida. So if we look at Radiance first, Radiance is fantastic because it has long stems, as you can see, which make for ease of harvesting. You'll notice that it has an incredible glossiness to the fruit, which is definitely a heritable trait that we select. It also has excellent pollination. You'll see very few pinch tips or cat face tips on this variety. And it colors very evenly, even under high tonal structures where a lot of the UV radiation is blocked. And this is a picture from this variety in southern Spain where all the production is under these high tunnels. So it's very adaptable for those reasons. So color, harvest efficiency, pollination, gloss are a couple of quality characteristics that are really standing out about this variety. And then you look at Sensation, very different kind of variety, a lighter color that does better in hot weather. This variety is a combination of very large fruit size, very high yield, but also very high sugars and excellent aromatics. It kind of blows the paradigm where we think of the best tasting strawberries are always the small dark ones. It's really not the case for this. And so this really shows some of the quality characteristics in kind of a high aroma, high flavor strawberry variety. So much so that a trademark was generated for the variety, and Florida growers are sort of experimenting with marketing the name of the trademark, if you will, at the consumer level, which is something that is usually not done with strawberries in the United States. You see it in Europe and some other places, but not in the United States. And then finally, Florida Beauty, which is the most recent release. And the cool quality traits about Beauty is that it has this excellent heart shape that you can see that is extremely consistent. It has a great red color. It has really high sugar content with a little more acid kick than Sensation, which some consumers prefer. And finally, it has fantastic rain resistance. Rain tolerance to the fruit is very important in Florida because of the fact that we do get rain in the winter and in the fall and in the early spring. And you can get an inch of rain on this variety and you can barely tell the difference
afterwards. So rain resistance is a quality characteristic in Florida that you really wouldn't even think about in many other countries around the world or other growing areas. Now, what makes these quality characteristics so challenging to breed for in Florida is our environment. So this is a picture of one of the freezes that we typically get during the winter. So not only do you have precipitation on the fruit, but you have overhead irrigation water for freeze protection during freezes in the middle of the winter that can impact fruit quality tremendously. This January, we were at 25 degrees in a freeze like this, and 2 weeks later, we hit a record high in February of 90 degrees. Okay? So the quality challenges when you go through that kind of variation within a single season is incredible. For those reasons, I think that Florida is actually a fantastic place to breed for quality, because the kind of variability and challenges of humidity and rain and freezes and heat that we get within a single season, I think selecting in that environment is one of the reasons why our varieties have worked in so many different growing regions around the world. I think that within season variability has something to do with the adaptability, if you will, of the varieties to other areas. So first, I want to talk about quality characteristics in terms of flavor, which is obviously what most people are most excited about with strawberry. This is my daughter Eve, who is 7 years old. As you can imagine, she has me wrapped around her little finger. Who wouldn't be with a cutie like that? But if you were a strawberry breeder and you're feeling bad about the flavor during a particular week, bring Eve with you to the field, because she will eat every strawberry and tell me how amazing it is. So she's my favorite taste tester in the world. She makes me feel good about my strawberries. However, her palate is not the most discriminating, okay? So that's where we need our taste panels. And in strawberry, taste panels are very, very important to us. So we do 2 different types of taste panels. When we narrow down to about a dozen selections in a given year, what we call our stage 3 or stage 4 trials, we have a trained taste panel. So we have about a dozen people whose palates are trained with a particular scale: caffeine for bitter, sucrose for sweet, hexanal for grassy, and on and on. And they give us very detailed information on about a dozen selections. When we narrow down to about 4 or 5 selections, we go to a large consumer panel where we feed 100 to 130 or 40 consumers in any given sitting a large number of strawberries of a smaller number of genotypes, and we ask them fewer questions. So for a consumer panel, we're mainly concerned with flavor intensity, sweetness intensity, texture, and then the most important of all, which is liking. How much do you like this strawberry? And we work with food scientists and also sensory experts from the dental school who created scales for these consumer panels that are fascinating. So for instance, what a panelist will do when they sit down is they will ask you to rate on a -100 to 100 scale what is the most intense experience you have ever had in your entire life. And you rate that on the scale, and then you eat the strawberries and rate the strawberries on the same scale. I don't understand how it all works, but it's fascinating, and it really gives us fantastic data. Now in strawberry, overall liking, as you can imagine, is very highly correlated with sweetness, okay? Flavor is also important. Texture is important. But sweetness is correlated at a high level with the overall liking. So sweetness has to be our main focus in strawberry flavor, or at least perceived sweetness, because perceived sweetness is certainly impacted by sugar, okay? So we have sucrose, fructose, and glucose, and those total sugars, as you can see from this graph, are going to be very highly correlated with sweetness ratings in our panels. However, what was interesting to us is that total sugars doesn't really explain all of the sweetness perception in strawberry. For instance, what is the difference between one of these points way above that red line and one of the points way below that red line? And I'll get to that in a minute when we start talking about volatiles. But first, I want to talk a little bit about sugars and the challenge with sugars in strawberry. So one of the challenges in strawberry, of course, is that though I breed for the consumer, there's a lot of intermediaries between me and the consumer, okay? There's the wholesaler, there's the retailer, there's the shipper, there's the fruit grower, and there's even the nursery grower who grows the plants for the fruit growers. And every one of these other parts of the market chain have different quality characteristics that they demand in a variety. So the consumer, of course, wants sweetness and internal color and a fresh appearance of the fruit. But the nursery grower wants good runner production for clonal multiplication. The wholesaler wants shelf life. The shipper wants size consistency, so the pack looks good. There are just so many characteristics, and the problem is that these characteristics interact at a genetic level. So let's take the case of sweetness, which is primarily driven by sugars, and yield, which is very, very important to the grower. Particularly in Florida, early yield is important because prices are highest in November and December when the supply on the U.S. market is very low. And yield and sweetness interact to a great degree in strawberry. In fact, the additive genetic correlation between early yield and sweetness is negative 0.76, okay? Incredibly strong. And I think one of the reasons we see the strength of this correlation is in Florida, and other breeders in other locations have not seen this strongly, is that in Florida we can have these hot nights and the plant is burning energy. And so I think that there's a basic energy relationship between fruit load and the amount of sugar that a plant can put in a given fruit. And so this is something that we have to watch very closely. This is a Monte Carlo simulation showing what this means in terms of this negative genetic correlation. So here we have, genetic, on the y-axis we have genetic gain, and on the x-axis we have the index coefficient for soluble solids content, which in strawberry is very highly correlated with total sugars, and it's what we usually measure. So if you look on the left side of the graph, if we ignore soluble solids completely and just select the best parents for early yield, we can have incredible genetic gain for early yield. This gets a breeder excited that I might be able to get 25% gain for early yield in one generation, but the problem is your solids will go down by 10%. If, on the other hand, we were to ignore early yield and we were to focus just on soluble solids, we could increase soluble solids by 10%, but our yield will go down by the same amount. So we're looking for an index that balances these traits somewhere where those lines cross in order to make some gradual progress for both traits at the same time. So index selection and balancing these two traits is very, very important for us in breeding. But the other question that this brings up to me is, what about sources of sweetness in strawberry that don't involve sugar, that may involve less energy output on behalf of the plant? And that's where we start looking at volatile compounds, because what is really fascinating is that the difference between, say, these two strawberry samples comes down largely to volatile content. That's one of the surprising things, is that volatiles in strawberry don't just affect the flavor rating of the consumer panelist. Normally we think of the volatiles as giving the, what differentiates a strawberry aroma from a blackberry aroma, from a raspberry aroma, and so forth. But there's actually a number of compounds in strawberry, apparently, that affect your perception of sweetness. In other words, if we have higher amounts of certain volatiles, we perceive the fruit from a sensory standpoint as having more sugar than it really does. And so, given that I'm a breeder trying to breed for both flavor and yield, this is really highly interesting to me. So as I mentioned in my question to Dr. Dudareva a little bit earlier, there are some very high abundance compounds that we just don't see much evidence for sensory relationships with those volatiles. But there are other volatiles with very low compound production, they're not abundant at all, that seem to have very high effects on flavor. One of these is a compound called gamma-decalactone, which is the peachy flavor that is actually highly abundant in peaches, which is in the Rosaceae family as well, by the way. And that very same compound is produced at high levels, well, not really high levels, but is produced at very detectable levels in some strawberries, but in half of commercial varieties worldwide, it's completely gone, completely absent. And what it comes down to is the deletion of a fatty acid desaturase gene that converts fatty acids to this compound. And so this was identified in some collaborative work with Kevin Folta, who's a fantastic collaborator at the University of Florida, and Alan Chambers, who was a student of ours, who's now a tropical fruit breeder at the University of Florida. And Alan used some RNA-seq data to establish that this gene, in fact, was the on-off switch for this volatile in strawberries. And so we were able to generate an HRM marker for this and now use this in marker-assisted selection. We've not found any negative associations with the presence of this trait, and we'd rather have it than not based on the sensory evidence that we have. So this is an example of where we're trying to look at these non-sugar-based sweetness enhancers and do something about them in breeding. This is an example of a volatile compound that's already present in cultivated germplasm. But introgressing new traits, particularly in flavor, is also very important to us. And one of the interesting compounds that we're highly interested in is one called methylantranolate, which you may be familiar with from some other crops. But methylantranolate is the characteristic essence of Concord grape from Concord grape, and is synthesized to put into grape Kool-Aid. So the grape Kool-Aid flavor is methylantranolate. And it so happens that methylantranolate is virtually absent from cultivated strawberries, but is highly present in the diploid Frais de Bois strawberries from Europe that are highly aromatic. And so it turns out that grape flavor, if you will, is actually a natural strawberry flavor. And so we've been able to introgress this into some of our strawberries, and this is very low abundance, but when people eat these strawberries, they say, wow, that's different, what is that? And usually most of them will eventually guess, oh, that has a grape essence, and it really tastes fantastic to some people, and other people say, that's not a strawberry. But I think there's a niche market there that's really interesting, because you feed it to some people and they start searching the rows for more. They cannot get enough of it. So we're excited to see where that goes. So, sort of in summary for flavor, yield sugar balance, quantitative genetics, index selection, and non-sugar sweetness enhancers from volatiles that we're trying to look at in terms of molecular breeding. So we're looking for more of these target traits in volatiles. We're continuing to do sensory work, and we'll see where that goes. But I also want to talk about another category of quality in Florida that's important, and that's disease resistance, because in our hot, humid environment in Florida, and with our environmental and variation, we have a lot of diseases that it can attack flowers and fruit, and so affect quality and yield very directly in strawberry. So here's just a few. Anthracnose fruit rot is a huge one. The resistance was mainly developed in Florida in the 1960s and 70s because of how prevalent this fungus is in Florida. But you've also got Phytophthora leather rot, which also attacks crowns and causes a crown rot, which is a big problem. And then you've got an angular leaf spot, which is a bacterial pathogen that affects the leaves, and also the calyx, the green top on the top of the strawberry, and can make the strawberry unmarketable. There's others as well, but I want to just give a little example from Phytophthora leather rot about what we're trying to do with disease resistance that affects quality. So one of the collaborations we have that has been really important in this is the Rose Breed project, which Rex has been involved with, and I'll highlight some of his contributions as well. But basically the development of tools, and some of these crops like strawberry that have been way behind in molecular tools in recent years. When I took over the strawberry breeding at the University of Florida, we didn't have a single marker for a single trait, okay? And today we're way ahead because of this project. So one of the very important tools that was developed was the iStraw 90 Axiom SNP array. So this array is full of subgenome specific SNPs that allow us to differentiate the 4 subgenomes of octoploid strawberry and allow us to map strawberry now as essentially a diploid with 28 linkage groups. This was an amazing step forward for us. And also, another characteristic of the rose breed is that they invested highly on this software called FlexQTL, which you've probably heard of before. But it's a fantastic software that we really enjoy because it allows us to do simultaneous QTL detection in many different crosses at once. And that's the kind of populations we have in strawberry. A lot of different crosses among a lot of different parents that are all pedigree connected. And so it gives us a lot of power for QTL detection in strawberry, and it's kind of like a mix between association mapping and traditional QTL mapping, if you will. So I want to just give an example of how we kind of combine this SNP array and FlexQTL to try to look at some of these disease resistance traits. So this is an example of a population that we recently did QTL detection in. So keeping with the theme of complexity, a highly complex population. So we have about 60 parents, over 100 families, a ton of different seedlings, but all highly connected in a mating design so that we can look at alleles in so many different genetic backgrounds at once as they segregate. And this is PC2, which is a major QTL that controls the resistance to the Phytophthora cactorum that I talked about that causes a crown rot and a leather rot of fruit. And one of the other cool things about FlexQTL is that it has a great haploblocking function. And so we discovered that in this QTL region, we actually had 3 different haploblocks. We were able to use that to hone in on which SNPs we really wanted to focus on in that region for SNP haplotyping. And we did some allele analysis, and we discovered that there were 4 major haplotypes, which was a fairly low number compared to what we predicted. And the effects of those haplotypes were very clear. We had 2 resistant alleles, apparently, each with different effects, one that was a little stronger than the other, and 2 susceptible haplotypes. And so it turns out when we look at our pedigrees, indeed, we do think we have 2 different resistance alleles. They come from 2 different sources and have 2 different levels of effects. And working with FlexQTL in that way really allowed us to parse that out and discover what we were dealing with. And so we ended up developing markers for each of those different haplotypes. And we're able to use that now in marker-assisted selection. And when we make a selection in a seedling population for the resistant alleles, we get up to about a 60% reduction in disease incidence from the use of this marker in selection. So a really cool success story in bringing something where we had 0 resources a few years ago to doing QTL detection and marker development. This is just a summary of the sort of quality and quality-related traits that we now have genetic locations to single subgenomes in the strawberry genome now. So gamma-decalactone is there on 3B. We have the switch between short day and day neutral strawberries. On 4B, we have a couple of Calototricum resistance loci on 6A, angular leaf spot, and then the Phytophthora on 7. So the fact that we can find so many genes linked to single subgenomes in strawberry has been great in terms of marker-assisted seedling selection. So in our strawberry breeding cycle, which originally had no presence of marker-assisted selection, we've included marker-assisted selection between the cross and the summer nursery now, and then of course for parent selection. And our breeding cycle is typically shortest possible, a 3-year cycle in annualized strawberry, where we have a summer nursery and seedling selection 1 year after the cross is made, and we test those strawberries in clonal plots as advanced selections the year after that, and then can use those in crossing about 3 years after from basically from seed to seed. And one very important characteristics of annual strawberry is that we have a summer nursery process where we clonally replicate each seedling before it's evaluated. And we only have a few weeks between germination of seed and sending those seed to the summer nursery for the next cycle. So our marker-assisted seedling selection has to be done in a pretty quick time frame. This breeding cycle feeds into the final topic that I want to talk about, and that is genomic selection for complex traits. So with Rex's help, we've incorporated genomic selection for parent selection in strawberry. We've found that Bayes bee models work very well in strawberry. We use about 9,000 SNPs typically for parent selection. And this is kind of how we do it. So in every year we have a clonal trial there at the bottom of advanced selections. That's usually about 300 to 400 entries in each of those trials. And we use those as our training population for the next cycle of advanced selections. And so we can predict forward a year in terms of clonal performance for a number of traits like soluble solids content, like fruit cull rate, which are a couple of very important quality components for us. And we predict forward, and what that allows us to do is take some of our seedlings that have never been tested in a clonal trial and move them into crossing 1 year before they would typically be done at the earliest stage. 3 is really the absolute earliest we can do that with clonal testing, because we usually clonal test multiple years before using strawberries as parents. And so we can build up those training populations over time, over multiple years, and so now our training populations are getting much larger. But this is what we do in parent selection. And here's an example of the selection efficiency we get, which has actually been very good. So with markers only, we can get about, for instance, with fruit size, about 70% of the gain that we can get with markers and phenotypes together. So it's working very well for us. And these gains are pretty indicative of the type of gains we get across other quality traits as well. Now, how does this work practically in terms of incorporating genomic selection for quality traits in strawberry breeding? Well, this is how we're doing it so far. What we're doing is, during the summer, between seasons, we use our marker data to predict breeding values for the individuals for which we only have marker data. And what we do during November, December is those individuals go into a clonal trial, and during the earliest part of the season, we can get some visual observations of those parents as well. So for other traits like color and shape and diseases and other things, we can get a couple months of field evaluation in for those traits, for non-objectively measured traits, if you will. And then we combine that information together to select a few new parents in each crossing cycle to use 1 year ahead of when they would normally be used in the crossing cycle. So essentially, we're sort of combining our visual observations with an early indication of parent potential for these objectively measured quality traits, and combining that together to make a few decisions about some additional parents to use. So that's how we're using it. We've been asking the question, how many markers do we really need in order to look at some other applications of genomic selection in the future? And for parent selection, we can easily get down to 1,000 markers for these traits. And that's for a large set of diverse advanced selections. And so we know we can go lower than that for within family selection applications. And so we've started to look at that, and with Rex's help, we've come up with some experiments where we're trying to rank seedlings within a cross using other half sibs. And we have about, for instance, in this mating design, 75 to 100 seedlings in each of these families. And what we've found is that there is some predictive ability that we can get out of this. It's lower than for our parent selection, but it does allow for us to get some genetic gains via markers. And if you look across traits, though the predictive abilities don't look that impressive, when you look at it on a genetic gain sense, we can get up to 15% gain over the family mean for some of these traits using markers. When you compare that with the fact that our gains for quality traits over the last 5 years have averaged between 0 and 4% gain per year, that's really fantastic. So we're looking at some reduced density marker methods that can hopefully be affordable to do this kind of application in the future. And the way we would envision doing that is doing our first selection with our major gene traits that we're interested in, and then using 500 or fewer SNPs to hopefully do a selection after that for our quantitative and complex quality traits. So that's how we hope that we might be able to do this in the next 2 or 3 years. So, just to summarize some main points for the talk. Flavor is so important for us, and we're using what we call sort of consumer-assisted selection to really use consumer sensory panels to tell us what our chemical targets are in strawberries and to make sure that we're accounting for our quality and our agronomic traits in our selection. Second, fruit disease resistance is a big focus of us because of the difficult environment we breed in, and we've just been so excited with the tools that we have to look at subgenomes in strawberry now that allow us to look at these major gene traits. And then finally, we're hoping to incorporate more and more genomic selection over time as we develop these seedling selection methods with genomic selection. And you know, as the phenomics world moves forward as well, we hope that there's going to be many, many other types of quantitative traits related to quality that we can look at. So I believe that my time is getting a little bit shorter here, so I just want to acknowledge the great team that I work with at the University of Florida. It really takes a big team of people, both in my group and outside of my group, who have been fantastic collaborators. So I really just want to acknowledge them, particularly Sujit Verma there at the top, who's our FlexQTL expert, Luis Osorio, who does our genomic selection work, Song-Hee Lee in the middle there, who does our HRM marker development and is working in a lot of molecular areas. Kevin Folto, of course, and then a bunch of other folks, pathologists, Rosebreed folks, Rex. Thank you all for your contributions. And with that, if I have time for questions, I'll be glad to take them.
Thank you, Dr. Whitaker. Questions for Dr. Whitaker?
One over there, I think. One there too? Okay, yeah.
Hello, Dr. Victor. Thanks for your great talk. And so you mentioned that one challenge for strawberry breeding and genetics is its ploidy level. So can you talk a little bit about what are the general strategies to overcome that challenge?
So the question was regarding ploidy level in strawberry and dealing with that. And since strawberry is an allo-octoploid, if we can get subgenome specific SNPs that only segregate in that particular subgenome, essentially we can map it as a diploid. And that has been essentially what we've been able to do. Out of the original 90,000 or so SNPs that were designed for that array, it turned out that almost half of those could be scored in a particular subgenome. But in terms of marker development, the subgenomes have been a big challenge because you get a lot of off-target amplification from homologous regions in other subgenomes. And so one of the fantastic tools that we've just gotten access to is an octoploid reference genome developed by Steve Knapp at the University of California, Davis. And they've been very kind to collaborate with us and give us some early access to that genome. And that's been wonderful because we've been able to identify polymorphisms between subgenomes that allow us to develop marker assays to target single subgenomes. And so that octoploid reference genome, that assembly is already working wonders for us. And in the next 2 or 3 years, we anticipate 3 different groups to come out with different octoploid reference genomes. So the community will soon have many more resources than we currently do. So a great question.
So we have time for one more question.
So my question is a little bit out of ignorance is,
How's strawberry harvested, by hand or mechanically? And then when they're harvested, are they fully mature at the time of harvest?
That's a great question. So strawberries are all hand harvested currently, and they do not ripen after harvest. So they're highly perishable, and in the United States, they're hand harvested straight into the clamshell, so they're only touched one time before they're bought at the supermarket.
So then, but it comes back to the reason I asked that is for the taste testing. How do you conduct that? Do you try to mimic that delay till it gets to consumer and shipping or not?
Yes, we do. So in sensory panels, what we do is we try to harvest them at sort of the commercial ripe stage, which is a little less ripe than the ideal stage if you don't have to ship them. Ideally, you let them ripen and they get soft, but they're more flavorful. And then we also cold store them overnight before we sample them, because we know that that's going to drive off some of the volatiles and it's going to affect other things. There's going to be some water loss and other things. So we try to mimic that a little bit to take that into account, because while in some regions for U-pick that wouldn't be relevant, for commercial strawberry production, that's absolutely what we have to do. Yeah.
Probably we can take one more question.
Time for one more?
Yep.
On your genomic selection applications, where do you need to be? Where's your price point for, marker cost per sample to really make it effective? And how do you balance that in strawberries with, time towards genetic gain versus cost?
That's a great question. For seedlings, one of the things about strawberry production is that it's very, very expensive. It's a high-risk endeavor. Strawberry growers in Florida have to gross almost $30,000 an acre to make a profit. So therefore, strawberry breeding is very expensive too, and even the summer nursery process is very expensive. So my initial thoughts are that we're targeting about a $10 per sample max price point in order for us to be able to economically do seedling selection with genomic selection. And it sounds high, but strawberries are so expensive to grow and propagate that that's actually an economic point for us. Less would be great, but we think that if we can get it down to around $10, which we've got some companies working with us to try to do that, we think that's pretty doable. So we hope we can make that happen. Yeah.
Okay, please put your hand together to thank Dr. Vetager.
All right. Next on the schedule, if you have your program, it might be good to get it out now so you can reference that as I talk about this. But first, we'd like to do a quick acknowledgement to our invited student speakers from other universities.
So we have 5 traveling speakers that are coming here to present to you guys during these breakout sessions, as well as featuring some of our own University of Minnesota students. But we'd just like to take a second to thank Matthew from Iowa State University, Puneet from University of Nebraska-Lincoln, Jessica from University of Florida, Sushin from University of Wisconsin-Madison and University of Minnesota, and Brent Murphy from University of Illinois for coming all the way here to speak and share their research with you guys.
So you can go to various breakout sessions, whichever session you're interested in. But the schedule is on the screen as well as your program. But the North Star Ballroom will be right here. So if you're interested in the high throughput technologies, you can sit tight. Room 202 is where the breeding quality driven crops session will be held, and room 202 is just right across the hall. And so we have a session going on in there as well. And then in room 110, which is just down the stairs, there'll be someone navigating you to find that room as well if you're interested in the molecular genetics session. If you have any questions, just find someone with a lanyard and they can direct you to the session that you want to attend. Thank you very much, and we'll now just move around to those sessions. Hello to our live stream people. We're now going to cut the live stream. Sorry, but we'll resume back at 2 o'clock.
Right, you're fine. Is Anna up first? Okay, no, that's perfect. She'll be here in like 1 minute.